Hi everyone, thank you for being here. Today I will talk about delayed post-hatch feeding alters intestinal stem cell activities and intestinal permeability in broiler chickens. So first, the delayed feeding chickens is present in industrial. Due to the long processes and increase the productivity, delayed feeding chickens is present in industrial. So the delayed feeding can up to 72 hours. So during the noise hatch machine hatch over time, vaccination, sex sorting, transportation, these kind of factors will increase the long processes for both chicken to receive their first feed. And previous study indicate that the delayed feeding broilers have negative effects on performance, such as lower body weight, shorter villi longs, and smaller surface area. So right now we know the delayed feeding can cause a negative effect on morphology, immunology, digestion, absorption in chicks. However, we don't know the differentiation, development, intestinal stem cells, homeostasis, long-term effect, or controversial barrier function in delayed feeding chickens. So our aim is to identify the delayed feeding effect on stem cell ability and permeability in chickens. In order to study this effect, we use delayed feeding chicken as our models. So the normal feeding access to feed after they hatch, while delayed feeding access to feed on day 3, which delayed feeding for 72 hours. So in order to study the acute effect, the short-term effect of delayed feeding, we take a alien sample from day 3 chickens and isolate a crib from day 3 chickens. To study the refeeding effect, we isolate the crib from day 4 chickens. And to see the long-term effect, we sample in the day 14 and day 21 chickens to take the alien sample. And we also isolate the crib on day 14 chickens. From the body weight and relative organ weight results, we can see that the delayed feeding reduced body weight and relative organ weight in chickens, similar to the previous study results, indicate our model is working. We also use HE staining for histology analysis, and we saw the same trend that villus height and crib depth is lower in the delayed feeding chicken. So when we saw the crib depth the crib is the stem cell area, so we thought that the stem cell might involve in these mechanisms. We saw the crib depth is significant decrease in delayed feeding chicken. Therefore, we also see the crib fission. So the crib fission means one crib split to the 2 crib. So we calculate the crib fission rate, and we can see from data that the delayed feeding chickens has lower crib fission rate, which indicates their crib is likely to spread to 2 crib. In order to estimate the stem cell play a role here or not, we identify the epithelial proliferation. So we use BRDU staining to identify the proliferating cells. So we can see from the data, the delayed feeding chicken has lower proliferating cells and epithelial cell migration, especially in the short-term event. We also use real-time PCR to identify the proliferation messenger RNA marker, pCNA, and we can see from the data that the pCNA is significant decrease in day 3 delayed feeding chicken, which match our BRDU staining results. And this result indicate that the proliferation in epithelial is lower in the delayed feeding chicken, especially the short term. From the proliferation data, we can know that the stem cell might have some effect in short-term delayed feeding event. To estimate the stem cell activity, we use a method called entry culture. So the entry culture means we isolate a crib which contains stem cell inside from animals' tissues, and we culture it in the plate with 3-dimensional matrigels so that they can form like a mini gut called entry inside the plate. And from the data, we can see that the delayed feeding chicken has lower ability for entry formation, especially day 3. And also the entry size is significant decrease in delayed feeding chickens. It's the same trend with expansion index. So the expansion of entry means the entry can become more and more. The increased number depends on the days they culture. So they culture longer, the number will increase more in normal feeding chickens, but not the delayed feeding chicken. So day 3 delayed feeding chicken cannot increase the entry number during culturing. However, the day 14 delayed feeding chickens can increase the number. So these all data all suggest that the delayed feeding suppress intestinal stem cell activity at day 3 and day 14. To better find a good marker for stem cell, we use the RNA scope to target the ORM4 messenger RNA, and we can see from pictures they are all stain the creep area, which is the stem cell relied on it. Further, we use real-time PCR to quantify the stem cell messenger RNA marker expression. So we can see from the data that ORM4 significant decrease in delayed feeding chicken, both short-term and long-term effect. This data suggests that the stem cell really play a role here, so we can see that the delayed feeding altered intestinal stem cell marker gene expression at day 3. Since the stem cell ability changed by delayed feeding, we use real-time PCR to identify the intestinal epithelial cell lineage allocation. We can see from the short-term effect that the lysosome C, which indicate the penny cells, increased in delayed feeding chicken, and the chick A, which represent the enteroendocrine cells, increased both short-term and long-term effect in delayed feeding chickens. The mucin-2 is the goblet cells marker, so we can see from the data in short term, the delayed feeding chicken decreased the mucin-2 messenger RNA expression. And we also use the ocean blue staining to quantify the goblet cells number in long-term day 21 chickens. And we can see from the data that the delayed feeding chickens increase the goblet cells numbers. So with this result, we can see that the delayed feeding alters intestinal epithelial cell lineage allocation. So their differentiation is different because the delayed feeding chicken, and that's in the short time. But this short time might cause the long-term effect in the future life. To see if this kind of short-term stem cell alternation affect the long-term effect, we determine the intestinal epithelial barrier function by using chamber. Briefly, we mount the ileum tissue on the chamber so that we can determine the electrophysiology on it and also do the physidatrine permeability assay. So from the data, we can see that the trans-epithelial resistance decrease in delayed feeding chicken in short term and long term, which indicate the chickens has leakier gut. So for the physidatrine permeability assay, we can see from the data that the day 14 delayed feeding chicken has a higher permeability. So this data suggests that the delayed feeding chicken has the long-term effect, which is more permeability and the leaky gut. To investigate the mechanism between delayed feeding and stem cell ability, we estimate the refitting effect on enteroid formation, size, and expansion. So we can see from the data that when the delayed feeding chicken refeed for 24 hours, the enteroid formation dramatically increased, but not the enteroid size and expansion. So the data here indicate that the refeeding can restore partially intestinal stem cell function. So we can see that the refeeding really do something to the stem cell ability. So we know that refeeding can restore partially intestinal stem cell ability. Then we are thinking, how about the film in intestinal, which is glutamine? So we are using the glutamine to add into enteroid culture media to see if there are any effect. From the data, we can see both the short-term and long-term effect. The glutamine can dramatically increase the enteroid formation, which indicate the stem cell ability. Although future study need to be done to see if the glutamine can restore the long-term effect in 72-hour delay feeding chickens, this data suggests this trend that the glutamine can restore partially intestinal stem cell function, just like refeeding. We can see from the conclusion figure, delay feeding chicken has a lot of short-term issues, and some of these issues can maintain to the long-term situation, which become the long-term issues. For example, the body weight is still lower compared to normal feeding chickens, and especially the functional parameter, for example, the gut permeability and the stem cell ability. So here we can see that the stem cell ability and gut permeability is both maintained to a long-term effect. So in this case, we can see that the stem cell ability is likely to highly related to the delay feeding in chickens. And this stem cell ability can be restored by refeeding and restored by glutamine. So future study will need to be done to figure out the detailed mechanism and also to see if the glutamine can help the 72-hour delay feeding chickens to restore their stem cell ability in vivo. I would like to thank everyone who helps to complete the project. Any questions?